Infectious diseases are bad news. As humans, we are constantly being exposed to pathogens like bacteria, fungi and viruses, some of which have the potential to destroy us. In order to defend ourselves, our bodies have developed barriers such as the skin, mucous membranes in the airways, and acid in the stomach. These barriers are able to prevent the vast majority of pathogens entering our body. But what happens when this first line of defence fails? That's when the immune system comes in, and we'll be learning about how it works in this video. If a pathogen gets past these barriers and enters the body, an immune response must be triggered. Different pathogens have differently shaped antigens on their surface. For example, the antigens on the surface of a flu virus will be different to the antigens present on HIV. The immune system is able to respond in a highly specific way to these antigens, but not immediately. We'll get to that in a minute. First, we start with a much less specific immune response. A type of white blood cell called a phagocyte is able to recognise cells that are not part of the body. They might detect a certain molecule found only in bacterial cell membranes, or a piece of viral RNA, for example. Once detected, the phagocyte engulfs the pathogen, trapping it inside a vacuole. Phagocytes contain lysosomes. These are vesicles that are full of digestive enzymes called lysozymes. The lysosomes fuse with the vacuole and the pathogen is digested. Finally, the pathogen's antigens are transported to the cell membrane of the phagocyte and presented. This allows the specific immune response to begin. The body has another type of white blood cell called a helper T cell. Different helper T cells have different receptors on their surface, and there is a specific receptor for pretty much any antigen we could ever come across in our lifetime. Once the right helper T cell has come across the antigen presented on the surface of the phagocyte, the T cell can bind to the antigen and is activated. The helper T cell releases chemicals called cytokines. Cytokines activate B cells and cytotoxic T cells. Cytotoxic T cells start killing any cells that are infected with the pathogen. When B cells are activated, they clone themselves in a process called clonal expansion, producing many plasma cells and also memory cells. Plasma cells make and release antibodies that are specific to the invading pathogen. They attach to the surface of the pathogen and destroy it, or they can clump several pathogens together so that a phagocyte can ingest large numbers at a time. Memory cells remain in the body for a long time. If the same pathogen enters again, the body will be able to produce the right antibodies much more quickly, destroying the pathogen before we even feel ill. Once we're able to do this, we say that we are immune to this pathogen. Let's have a look at an antibody in more detail. Antibodies are made of 4 protein chains that are held together by disulfide bridges. As you can see, there are 2 much longer protein chains called heavy chains and 2 shorter protein chains which are called the light chains. The blue region, called the constant region, is the same in all antibodies. The variable regions differ and are very specific to one particular antigen of one particular pathogen. This makes antibodies extremely specific. So that's it for this video. I'll put up a slide with a summary of the immune response, and you can check your understanding by seeing if you can talk through all the steps. There's also a printable worksheet in the description. Please like and subscribe, and I'll see you in another video. Thanks for watching.